For sequences that are to be cloning vectors for expression of recombinant proteins, generating mutants, or sequencing, one of the most important techniques is the PCR amplification. Transformation is the kinetic alteration of a cell resulting from the direct uptake and incorporation of exogenous genetic material. Genetic engineering for this process are used plasmids that are DNA molecules that automatically replicate and can be used as a receptor of foreign DNA fragments, and they are most commonly used vector. Select according to the DNA size to be inserted in the ligation proposed, with help of enzymes. One cloning system is TA that uses topoisomerase does not require restriction enzymes. The procedure exploits the terminal transferase activity of a certain thermophilic DNA polymerase, including Thermophilus aquaticus or Taq polymerase. But in this protocol is used the topoisomerase 1 of the smallpox vaccine virus. This because it's being a double-strand DNA, especially in the sequence CCC-TT, leaving a French mean. But with the interaction of the adenine of the PCR product, the bond is destabilized and forms a new ligation. The PCR 2.1 topoplasmid has the beta-galactosidase enzyme as a selection marker by color distinction, and its reading frame is interrupted when the PCR product is inserted, and the colony expresses color white. But if the PCR product isn't inserted, the beta-galactosidase gene is expressed.
The first step is to out the PCR product, the PCR 2.1 topo, and the other reactives using ice. Then, prepare the ligation reaction as can be seen in Table 1 for the chemical transformation. Incubate the reaction at room temperature for 10 minutes. After that, transform 3 microliters of ligation reaction in 50 microliters of chemiocompetent cells. After the transformation, add 300 microliters of SOC medium and incubate at 37 degrees with stirring during an hour. Subsequently, spatulate ampicillin and exgal in a plate with LV medium, and finally, sew the competent cells into a LV plate with ampicillin and exgal. In the petri dish, only 3 colonies grew. None of them show expression of exgal, which means that the plasmid was not successfully inserted in the cells. For cloning PCR products directly from a PCR reaction in just 5 minutes, a PCR topo vector with covalently bound topoisomerase 1 for fast cloning and recombinance is used. The vector includes 3T overhands for direct ligation of Taq amplified PCR products, T7 promoter for in vitro RNA transcription and sequencing. The vector also contains M13 forward and reverse primer sites for sequencing. Echo R1 sites flanking the PCR product insertion site for easy incision of inserts. Kanamycin and ampicillin resistant genes. Easy blue-white colony screening for selection of recombinants. The elements that conform the vector PCR 2.1 topo are the following. Replication origin, specifically the vector topo TA cloning kit PCR 2.1 topo consists on 11 regions GATC in a 254 BP fragment. This origin of replication is known as ORIC3. Resistant gene. E. coli acquires resistance to selective media, kanamycin and ampicillin resistance, and can grow smoothly. Multiple cloning site. It has a unique targets for specific restriction enzymes and allows to introduce new cDNA fragment. Additionally to the PCR 2.1 topo vector, there are another variants from the topo family vectors. For example, PCR2 topo, vector. This vector has the same characteristics that the vector PCR 2.1 topo, but has two different promoters, T7 and SP6 for in vitro RNA transcription and sequencing. To verify the ligation in the topo vector, the following methodologies can be used. PCR reaction. Using initiation and depending on the size of the product obtained, the cloning of insert is confirmed, taking an amount of the colonies and then programming the PCR with the following conditions. Verification by electrophoresis. Run an electrophoresis gel with the cloned vector to observe the size and compare it with the vector without the ligation. Enzyme digestion. Perform a cut in the site or sites in which the insert was placed, then run an electrophoresis gel to see how many bands appear.
There are two types of white and blue colonies were obtained, selected by resistant to ampicillin, since both types of bacteria carry the plasmid one. The blue colonies correspond to the cell transfer with the vector that takes the gene of beef galactosidase functional and produced by induction with EPTG, this enzyme able to hydrolyze to the X-gall and to generate color blue. The white colonies correspond to the cell transfer with the vector that takes gene of B galactosidase non-functional by insertion of a fragment of DNA within himself. In order to be able to introduce our DNA fragment in the bacterium vector is needed collation. This one is not more than non-genomic DNA sequence, PCR topo. The union of the sequence the vector is carried out thanks to the topoisomerase. There are special considerations for the clonation of PCR products. In the first place, an aliquot of the PCR reaction is due to analyze in agarose gel before being used in the ligation reaction to verify that a single product of PCR of the sustainable site is had. Later, it is desired to come to its purification. This step can take to using diverse techniques, like the classic upgrading phenol chloroform, either recent others that more that use as columns provide with special filters of resin of its cellulose that they unit in nucleic acids. Once purified the PCR product, the ligation with the vector can become to the reaction to form new recombinant molecule. The best results are obtained with the relation to mole of inserted ligation vector is 3 to 1.